Like Pedro, there are many people every year that are diagnosed with osteoarthritis. Osteoarthritis is a degenerative disease that is characterized by the wear down of protective cartilage within the joints over time. Many patients with this disease eventually require a joint replacement at some moment in their life, which fails to restore long-term physiological tissue function. In 2023, about 3,715,320 joint replacements were performed. This current data shows an active increase in the number of patients that require any type of joint replacement in the US and worldwide. In order to help address this problem, our team is designing and manufacturing a fully operative bioreactor capable of differentiating stem cells into cartilage cells, or chondrocytes. It will be used by lab research assistants, scientists, and surgeons to study varying growth conditions. The bioreactor will operate up to 3 weeks on a standalone, sterile, and self-feeding basis. It exposes the stem cells to precisely regulated axial and shear compressive forces while maintaining an optimal cell culture environment. We are Team 26006, and this is DeBeck. The Dynamic Bioreactor for Engineered Cartilage, or DBEC, is a system that will be used to research how different loading forces and loading patterns affect chondrocyte development during in vitro culture, with the ultimate aim of producing engineered cartilage that mimics the histological and mechanical properties of native tissues. This is the DBEC system block diagram. It is composed of multiple integrated subsystems, which include the automatic feeding system, incubation chamber, mechanical loading system, and graphical user interface.
The feed-through system is a novel addition to many pre-existing projects of this sort. Our feed-through pipes take media from a refrigerated system and autonomously transport it to each well inside our incubation chamber. The media passes through roughly 2 meters of tubing where it is heated to 37 Celsius in the span of 20 minutes. Flow is driven by 6 individual micro-peristaltic pumps that move media at about 1 milliliter per minute. This whole process maintains sterility through the use of compression bulkheads at interfaces and pumps that never touch the media. The media is then extracted using a vacuum trap system. To make the system autonomous, we inserted a solenoid valve that opens and closes the vacuum line through an electronic signal. We also use a custom converter that joins 6 tubes to a single tube for simultaneous extraction of media in each well. In order to fulfill system requirement 2.7, we tested all 6 pumps on a single DC voltage source. We found a linear relationship between voltage and flow rate, which we used to fill each well to a user-specified volume. To test our extraction system requirement 2.8, we used a vacuum trap system given to us at the lab, and we were able to extract all media within 2 minutes from each well.
Our cooling chamber stores fresh media for the system to use over the span of 3 weeks. We create a polycarbonate shell that holds polyurethane foam that serves as an insulator. Here, the media is stored in a lab standard 2-liter glass bottle with a custom cap. The custom cap has 6 holes for pipes to exit the bottle and move into our incubator. The cooling chamber also has 6 drill holes through its body to allow for the feeding system to pass through. This helps satisfy system requirements 2.7.
The mechanical loading system is designed to deliver physiologically relevant stimuli that mimic the natural environment of a joint. Our system utilizes a biaxial loading system, applying both compression and shear strain to replicate in vivo mechanical conditions. This is achieved using linear actuators that drive motion along each axis and apply force to plates contacting the well plate, ensuring uniform and controlled loading. Because the compressive components directly contact the hydrogel scaffolds, we incorporated custom silicone caps on the contact points to prevent damage and maintain biocompatible conditions. To verify the mechanical loading system, we needed to ensure that our motors used had the correct torque to maintain the desired force and the correct step size for the resolution of strain required. From basic principles, we noted the maximum torque needed is 7.47 times 10 to the negative 7 Newton meters, and the step size desired is 1 micrometer. The NEMA 23 motors, along with the DM556T drivers, are able to sustain this torque and reach a resolution of 0.78 micrometers per step, fulfilling both requirements.
All parts are integrated with a user-friendly graphical user interface, or GUI, that controls and keeps in ideal environments for cell growth, as well as controls the forces applied and force patterns on the cell scaffolds. The modular software design creates a Python-based GUI where the user will set their experimental conditions, including temperature, CO2, humidity, and motor strain. The GUI will send this information over to a microcontroller that will parse the data and control the hardware. The microcontroller will facilitate the communication of data back to the GUI every 2 seconds, allowing for real-time system monitoring on the GUI screen and a CSV output file. The software controlling the autonomous feeding system was tested through 35 tests categorized in 7 classes using the PyTest testing framework. The main categories include Test Standard Exchange Cycle, which verifies the process for this feeding cycle. It verifies that the solenoid valves open, the pumps turn on, the solenoid valves close, and the pumps turn off in the correct order. Test Stop button and Test Reset button confirm that the stop and restart buttons on the GUI main window turn off the pumps and close the solenoid valves controlling the vacuum, as well as clear all data manager and internal signaling data. Test Pump Duration Calculation verifies that the formulas and internal conversions for the pumping cycle duration are mathematically correct. The software passes all of these tests with 100% scores. This result demonstrates that the software is sufficiently robust to direct the operation of the autonomous feeding system. We have similar Python tests in place for verifying each other subsystem software.
With the goal of advancing tissue-engineered cartilage to lower animal studies and clinical trials, sterility is an essential requirement for our systems. The FDA and engineering standards such as CFR 1271 provide strict guidelines for sterility in medical products. We strategically choose our products and materials for the incubation chamber connection holes to prevent contaminations by bacteria or viruses. Static connections for the inner box are bulkheads made of fully autoclavable materials, including PVDF and silicon. Static connections for the outer box are silicon grommets, which, again, are fully autoclavable. For dynamic motor connections on the inner and outer boxes, we utilize compressible bellows made of neoprene. These bellows allow the motor actuated rods to move in and out of the sterile chamber without introducing any new air or particle matter. We also consider the sterility requirements when choosing a material for our inner box incubation chamber parts. And after extensive research and discussion with the sponsor, we choose to fabricate the inner box, the wall plate, the water bath, and a transducer from a 316 stainless steel. Stainless steel is corrosion resistant and commonly used for medical applications. It's also thick enough to withstand the vacuum pressure inside the incubation chamber. For design day, we're going to do a sterility test by putting an egg or plate in our incubation chamber and then wait for 24 hours and then going back and check if there is any bacteria growth.
The main purpose of DBEC is to contribute to the research and development of orthopedic research in order to deepen our understanding of engineered cartilage and translate it into clinical settings in order to treat osteoarthritis.